So I'm very pleased to welcome Sasha Gorbalenya back to the Coronavirus Multi-Species Reading Group. You've really offered a delightful selection of your life's work for us to discuss today. We've got the piece, Could We Live in Peace with Viruses, as maybe the main guide and the one that's perhaps the most accessible to people outside of the field. But you've also offered us a very rich account of, where coronavirus understandings of both the basic gene sequencing, but also the bioinformatics, like how to make sense of this big question. So what is a virus is one of the questions you raise in this more popularly oriented piece. And I think all the things that you've offered us to read this week speak back to that. One of the things, and I've got a bunch of like super technical nerdy questions about the more bioinformatics, genomics articles, but one that we've already discussed in the context of Roberta's essay is this very interesting fact. For starters, we're all infected by viruses, and most of these viruses are non-pathogenic. But even for the ones that cause disease, you note in this essay, only a minority suffer. And in this essay, you ask, we may not escape the question what these infections cause, particularly in the long term, to others, to the majority who were infected, but who did not suffer noticeably. And I think this is a very interesting question in the context of SARS-CoV-2. One of the things that we see is that the majority doesn't get hospitalized, doesn't get put on a ventilator. And I think one of the important things that you've insisted as we've talked over the past few months is the distinction between the virus, SARS-CoV-2, and the disease, COVID-19. So now that you have, you've been thinking about this question maybe for more than 10 years since this lecture in 2011, how do you think about this question now, the minority suffering while the majority harbor these viral infections but don't get noticeably sick?
Yeah. Thank you, Eben. Yeah, it's of course this is a central question to interaction between humans and viruses, but probably this question not often asked because maybe answers are not very pleasant, and also at the basis of this question, basically, it is negotiation or compromises we make, in human society. So how you reconcile interest of majority and minority, right? And especially when the question is about death, even or suffering. So how you weigh then what you prepared to sacrifice as society, as a group. And it is very fundamental questions. So I think that simply this pandemic opportunity to ask this question again, but again, it's requires certain type of courage probably to discuss this question, especially in the pandemic when certain people suffer. And so answer probably would be, you have to make everything to save people, right? Regardless of the cost.
Mm-hmm. And so you provided Rachel and I this beautiful history of coronavirus research in an email as you introduced these articles for this week. And I might just try to summarize that for some other people who didn't have the privilege of reading the email. You talked about the sequencing of the first coronavirus genome by a team in the UK, focusing on avian infectious bronchitis virus. That's one of the papers that we have on the deck to discuss this week. And then a few years later, you were applying some bioinformatics techniques to really understand, not just letter by letter, line by line, what the sequences actually were, but how you might understand them as enzymes that actually do things inside of the cell. And then the 2003 paper that you offered us, I guess the key thing that we're looking at there is how coronaviruses replicate. And you're looking at a section to get technical. We're talking about open reading frame 1, and I guess open, actually I'll get technical later because I've got better notes somewhere else. And then you also offer us this paper of a planarian coronavirus genome, which is some of your more recent work. So maybe just going back to the 80s to situate, the emerging knowledge that you had then. At a certain point, all we knew, we had basic structures of viruses. We had coronavirus, electron micrographs, so we kind of knew what they looked like, but we didn't really know at that point until you were working on some of this work, what they actually did after they interfaced with the cell. So I was wondering if you could just walk us back through some of those findings, if you can take us back to this paper that you published with colleagues back in the day, the coronavirus genome, looking at the non-structural polyprotein by comparative amino acid, amino acid sequence analysis. So you're applying some of the very first computer-based modeling to understand how this particular genome related to others. So what did we know in 1989?
Yeah, yeah, this is of course was exciting times. almost 35 years ago, 80s, when genomics entered virology. As you know, genomics was not part of human studies yet and bacterial studies. So virology was first where genomics entered in 80s. And this was extremely exciting times because we learned about viruses from genomic perspective, which was very difficult to do was by genetics. We knew something about by genetics at that time. And there was a lot of discoveries at times, of course, from genome sequences. So this IBV genome sequence was the first of the coronavirus. And it was because this virus caused, of course, trouble in the poultry industry. So this is not by coincidence that this virus was sequenced first, not human. And this is also very telling because human infections by coronaviruses were not in the spotlight. It was poultry which was a driving force in the coronavirus. So this is very interesting. So, but when people sequenced full genomes, it was discovered how large the genome was. So people suspected that it would be very large, but it was too large to surprise the scientists. So it turned out that 70% or 65% of genome encode something that we did not know. We knew nothing basically about this. So what happening in the side cells was encoded by this part of genomes. And there was no knowledge about this. And it happened that I and my colleagues in Moscow at the time, we studied genome sequences by comparative genomics, all virus genome sequences. So at that time it was possible to analyze all virus genomes because they were few. And so, of course, we started analysis of this genome when it was released from the first days. And we were able to map major players responsible for replication of virus inside cells. And later it was confirmed that most of this analysis was correct, but it took 10, 15 years of experimental work to do this.
And maybe we'll jump ahead in time to one of your papers where this is, some of the details are starting to be confirmed. So, here you're dealing with a coronavirus that is very, very similar to the same species, in fact, as the one that's causing the pandemic. So SARS-CoV proteins, the 2003 paper with Eric Schneider, and you're identifying, I guess, 16 subunits of this viral proteinase and a total of 28 SARS-CoV-2 proteins. So we're going to get into some technical things here. And so for starters, maybe could you explain where the idea of an ORF came from, the open reading frame, because this seems to be like the key unit that people are using as they're trying to understand and parse these coronavirus genomes.
Okay, so it is even indeed very basics of genome sequencing. So when people sequence genomes, so they define basically order of letters. And of course, it's chemical molecule, but we translate this chemical molecule in simply just letter using for letters code vocabulary for nucleotides. But then you have to, but that is kind of a text, which is again translated inside cells in proteins. And so there is a table of conversion, how this code, nucleotide code, could be translated in amino acids. And so this translation is possible only in under certain rules, how you translate, you read this genetic message. And so where it's possible to translate is called open reading frame. So because it's possible 3 reading frames, so actually 6 reading frames theoretically possible in genome sequences, but only some of them actually can be translated in amino acids. And so these pieces and registers, they named open reading frame. It's open, it means that it can be translated. It's open. Yeah, and so it is done automatically because it is very simple rules. It is as I said, it was done by these rules were established early in '60s. So, but what is not easy to do, it is assign certain certain function to part of these open reading frames, which is will be proteins or domains, and that is what was done in this paper, also based on the our previous analysis of 80s and or in 90s of other coronaviruses, but analysis of SARS coronavirus was expanded in this respect. So it was used results of the previous analysis, but also introduced some new findings which were quite key for understanding SARS-CoV-2, SARS-CoV, and also all other coronaviruses. So of course, I could kind of discuss details, but I am afraid that it would be really kind of specialized knowledge. Yeah, so to probably I will stop at this point.
Well, this is helping me a lot, and I've taken the liberty of sharing my screen. Does the figure show up on your screen of the genome? Does that show up for everybody? So this is a figure taken from your 2003 paper, showing at that point the 28 different SARS-CoV proteins in here. And as I understand it, the paper is mostly concerned with the replicase/transcriptase. So this is basically how the virus makes more copies of itself. And all of these different proteins here, 1 through 16, are part of the way that it's replicating. Is that more or less a good description of this part of the genome?
Yeah, and so that was deduced by comparative genomics. So everything which you could see here, was simply just done by computer analysis. But of course, it utilized knowledge of other coronaviruses. And so it's basically project knowledge of other coronaviruses and also from other viruses. And in this way, it is similar result compared like was in the first paper with IBV in 80s. So that paper established a blueprint, basically how to do this. And it first time described this type of organization. And what was amazing, there was no experimental knowledge at all about coronaviruses in this respect at that time. So it was completely it was deduced from knowledge of other viruses, not coronaviruses. And so that was quite risky, I would say. But turned out it was correct.
And these structural and accessory proteins, some of these, like S, will be familiar to most people. Spike gets a lot of media attention these days. And then I took another figure that from this essay that shows some of the other proteins that were identified as of 2003. So the envelope protein, the membrane protein, the nucleocaspid protein, and I guess some of these are conserved across coronaviruses. I understand that part of what gives some people, that makes them not going back to that question, why does the majority not suffer from a viral infection? Perhaps there's enough similarities between the nucleocaspid protein on the surface, not spike, but other things that generate antibodies. Is that a pretty fair understanding of the basics here?
I mean, it's correct in terms of similarities and commonalities between all coronaviruses. All coronaviruses have these proteins, 4 proteins: spike, S, E, M, and N. So, and they form virions. And so they were first recognized even before genomics, because virions were available as particles and they could analyze by biochemical techniques. But, and it was easy to map them on genome because of this. But in respect of why some people, some animals suffer and others not, it is not easy to connect to this type of kind of basic picture. So this still we don't understand, because it's not it's related to virus, but it is related to subjects who infected. So it is people who suffer and who die. So most answer to this question, it is with people, not with virus.
And that's where maybe some of us can help, come up with those answers. We had some fabulous presentations early on, from everyone from Tony Hatch to Hannah Landecker, looking at how intergenerational historical legacies contribute to vulnerability to disease, and how those intergenerational experiences of things like racism and inequality can actually result in cellular level differences and, say, expression of ACE2. Back to some of this here, there's a lot of question marks in this 2003 paper. It's been about 20 years. Have you seen any of these unknown open reading frames? So this is the ORF number for people who are, just starting to get your heads around like these different parts of the genome. So do we know, for example, 3A, 3B, what these do now, or any of these other bits?
Yeah, of course, people focused on these proteins. So open reading frames encode proteins, and so proteins have the same names, like 3A, 3B. And these proteins, they called accessory proteins, because if you delete these open reading frames, virus still replicate in infected cells in model system. But for infection in population, virus probably needs these proteins. Otherwise, they would not have been there. And so what people learned, so mostly or exclusively, this open grading frame encodes a certain function which modulate interaction between virus and host. So in this case, humans or bats, for instance. And so it is kind of, it could counteract a certain defensive system in cell. So for instance.
So this is super helpful and enlightening. And then the other paper that you've offered is the largest, am I right, the largest RNA viral genome described to date, which is a coronavirus found in planaria. Is that still the case?
Yes, so it is not coronavirus, but it is nidovirus, because coronavirus belong to larger group, which is called nidoviruses, which share certain properties, and because of this, it's united and together. And so there are currently 14 families, 14 families recognized of nidoviruses, and coronavirus is only one family out of 14. And so one of these families includes this planarian virus, and which is the largest RNA virus in the world. Yeah.
And one of the things I got out of the paper is that there's an enzyme that does proofreading, which seems to be remarkable. So it seems like not only is this RNA virus replicating itself, but it's also going back to make sure that there's not too egregious, errors, that the mutational load isn't too high. So it seems like, many people think of viruses as just like piggybacking on cellular machinery to kind of do their thing, but a lot of the machinery is actually built into this this small set of proteins, relatively small compared to a eukaryote.
Right. Yeah. So yeah, absolutely right. Yeah. And so that was one of actually excitements which when people sequenced the first coronavirus genome, and turned out it to be so large. So it is not kind of primitive system. You have simply just a RNA polymerase, for instance, and capsid, and that's it. So you have a really complex genome. And so, of course, question was what is this about? And so one of the driving forces in studies of coronaviruses was understanding how this large genome emerged and what is about this large genome from the first genome sequence of 30 years. And this is what also was of my driving forces in my research was. And discovery of SARS coronavirus, which was first pandemic 17 years ago. Of course, it was devastating, but it was actually sequencing of the coronavirus, SARS coronavirus genome, prompted discovery of proofreading. So in this paper, 2003, was first time proofreading enzyme was identified. So in my research. And so that was the biggest surprise. And also it offered explanation why coronaviruses have a so large genome, because they can proofread, and so they could expand. And so that was a major discovery, and also because other viruses have, RNA viruses have smaller genome, and it explains many things about coronaviruses and also nidovirus.
And SARS-CoV-2 also has that proofreading open reading frame?
So all nidoviruses with genome size larger than 20 kilobases have this enzyme. So you need this enzyme to have a large genome. So there is no exception.
And it seems like a lot of these other ORFs are proteases. They're just chopping up so the big chains of protein get made, and then the proteases break them up into functional units. Is that more or less how it goes?
Right. And so here we, of course, have another amazing discovery with viruses, which was made in 60s, back when people sequenced, even did not sequence, but they studied poliovirus. And so if you maybe remember, so in 50s, outbreak of polio epidemic was something like comparable with what we experience now with SARS-CoV-2. And so much of research was directed to analysis of the cause of this outbreak, virus of poliomyelitis. And so people in 60s shows that in molecular biology of polymyelitis contradicts major dogma of molecular biology. Then one gene, one protein. So before it was a paradigm. So you have one gene and one protein. And turned out that when people studied polymyelitis, polymyelitis, that protein is produced, but then it's after catalytically, so it's intrinsically split in many other proteins. So 12 proteins. So it was violation of paradigm, and it provided explanation and also blueprint for many other viruses, including coronaviruses, because it produced huge protein, something like 8,000 amino acids. And it's important to understand that average protein in cell is 300 amino acids. So it's 20 times larger. So how this protein can exist? So in fact, it doesn't exist. It's cleaved after catalytically, so that it means it's self-cleaved. So it includes proteases inside, and they regulate, and so they cleave in the pieces which form replicative complex. So it is very intrinsic and a complex machinery, and the order of proteolytic processing and place extremely important for how these infections will go inside cell. It is not simply just mechanistically done. It is in time and space highly regulated.
So I'm peppering you with a million technical questions, and I hope other people start to jump in. So again, feel free to just unmute yourself. Is there already a question in the chat? Okay, Tom, do you want to unmute and ask that question?
Yeah. Hi. Yeah, I was just wondering, because in the context of human genome and eukaryote genomes, we talk about non-coding sequences and their relevance and importance. And I was wondering if viruses have no coding sequences other than transcription binding sequences and stuff like that. So.
Yeah, yeah, it's a very important question. So in human genomes, most of the genome is non-coding. So these open coding frames comprise only very small fraction. But in RNA viruses, it's opposite is true. 95% of genome is open coding frames. So basically it is genome which encodes proteins, but still something like 5% it's non-coding, and it's regulating sequences. So they regulate genome replication and also genome expression. And expression means transcription and translation. And probably in some cases also encapsulation, because genome must be encapsulated in virions. When virions form, genome must enter. And so it means that proteins of virions must recognize the genome, and they must select it from many other RNAs which is available in cells. And so in some cases, it is done through interaction with non-coding regions.
So can you think of the reasoning why viruses would not benefit from having longer non-coding sequences?
So genome has is a text, and so it is information, and so infection is mediated by proteins, and proteins are very versatile molecules, can do many things, but in some cases you don't need proteins. And RNA, a non-coding part, which doesn't encode protein, so it's still functional. So also there is a kind of major hypothesis that our life started from RNA, and so at the beginning RNA was non-coding generally, so it was able to do everything, replicate, and so propagate and everything. And so then proteins emerged, and so RNA was able to delegate certain certain function to proteins. That life became more complex, but certain certain function is still done by RNA and pretty well. And so that is the reason why we have the non-coding regions.
Interesting, thank you.
Now certain functions are delegated to humans and planaria and other host organisms.
Right. Yeah. So yeah, you could see some parallels, of course, but of course we could speculate only what happened before, kind of in the primitive system. But major principle could be the same. So complexity emerged, and so then division of labor emerged. But also there are certain constraints on the how complexity organized. So it's very general, of course, area, and you could kind of only speculate.
I have a protein modeling question, but wait, I see there's another chat. Okay, Soraya's got a question. I'll save my protein modeling question.
Can I ask my question too?
Oh, sure. I didn't see you, Astrid.
I posted a while ago. It's
Oh, a while. Oh, I didn't see it. Okay, Astrid goes first.
Thank you. It relates back to your non-technical paper, if I may. And the question is historical. I'm interested in what viruses are. And you mentioned 4 perspectives. How does what viruses are, or what do you think, how does that relate to how they are determined? So you mentioned this at the beginning of the paper, you speak about this very simple and ingenious test that all of a sudden rendered the invisible viruses visible. So I'm curious what this test was, and what kind of virus was, or what kind of entity was made visible through that test?
If I correctly, yes, thank you for your question. So if I correctly understood, so you ask how first virus was made visible, or yeah. So how actually virology was found. So because that is what's a question about what happened 100 in 1892 paper was published. So a virus is not the only small creatures. Of course, we have bacteria, right? So it's microbes. So all this micro life, which is very small, and so it's invisible by naked eye. So you need special techniques to do this. And so people discovered microbes using filters. So they have filters on the physical size, and you could make filters with these different pores of different size, and so and then you could kind of put something on these filters and then study what can get through properties. So in this case, and with microbes, of course, property which people were interested in, pathogenicity. So you see certain type of disease. So in this case, in plants, some kind of dots or something, so the plants suffer. And so you take extract from plants, put through the filter, and so then uses juice, and so put on another plant, healthy plant, and then you see that this healthy plant become sick. So that it means that property could be get through the filter. So it is very minute, and so you could propagate this. And so this is how viruses were discovered. They were discovered in plants first.
So that they were discovered by what they did to other organisms, basically, right. What they did to plants, and so they were assumed to be particles. So because they were filters of specific sizes. Right.
Right. Yeah. It's and so if you kind of look how virus is translated, if I'm not mistaken, it is Latin word. and it is basically translated in English like poison. So, and so this is what is a connection to pathogenicity. It is intrinsic with viruses. So, because this is how they discovered, so how people look for this, and this is how people perceive this, because it's like a mystery. Life is kind of gone, everything is fine. It's suddenly some people start suffering without any visible reason. So there is a kind of sunset, sunrise, everything, weather is fine, so everything is fine, but people got sick. And so that is because it's something invisible is going around. So that is why there is a deep perception of viruses and association with disease, because it is the only way we see it, unless you're scientist, and now you could kind of, you trust sequences of electron microscopy.
Thank you very much.
Soraya, and then Nina's also got a question.
Yeah, thank you. I actually have many questions, but I will try. You know, what I realized reading your papers is with the SARS pandemic. I mean, why hasn't that become this worldwide pandemic, right? Why did it stop? But that's not really my question. I have another question. I mean, I can see that with genomics, it's very powerful, you know, for the classification of these viruses and to follow to understand all the molecular mechanisms and also to understand how they evolve, right? But I wonder, to understand better this ecological function of viruses that we need to understand, right? So do you think that genetics is the right, or genomics is the right tool, or what do we need, or what kind of tools do we need to understand this ecological, which you tell us we should understand better, right? The ecological role of viruses, all the viruses we are not aware of because they don't produce diseases or something. So how do we get to those? I can see that genomics can help a bit, right? But to really understand the ecological part of it, I'm not quite sure. I would like to hear what you, so my question in a way, what does, genomics is a powerful tool, but what does it hide, you know? Or also, you know, we become more and more critical what we can actually see in the one-dimensional sequence of DNA, right? For at least for higher organisms, but maybe the story with the virus is different.
Right. Yeah. Yeah. It is this question. It's important, and also actually it leads to many other questions. Of course, this is important. But first, I just kind of remark about what you asked about SARS. So there are two characteristics which are important: disease, lethality, and transmission. And so these characteristics are fundamental for both viruses: how little virus and how fast it transmitted. So they are kind of independent. And SARS coronavirus was highly lethal virus, but poorly transmitted. And that is the reason why the first epidemic was easy to stop. And so because it was clear what is happening, but the transmission is very kind of slow. With SARS coronavirus 2, it is completely different picture. It is not lethal, but it's highly transmittable. And with genomics, so that is the tool we have to characterize viruses. And the only tool we have which can characterize viruses in unbiased manner. So we don't have other tools. So 15 years ago, there was no question, even when SARS epidemic happened 17 years ago, we did not have the tools at this scale at that time. Still, virus discovery was mostly through pathogenicity. So pathogen emerged, and so then you can study this. Nowadays, it's completely different. You have environmental sample, you sequence everything, and you're saying, okay, so there are many viruses and new viruses there. But what they do, what their roles, we have no idea. We don't even know who they infected. And you're absolutely right. So we need other techniques, and as scientists say, assays, to measure a contribution of viruses to interactions in the ecosystems. But we don't have these assays because you need a very complex assays because it is complex system. And to measure contribution of particular viruses, it is kind of quite challenging. Although people try to do this in isolated systems, but on the global scale, it is a hugely challenging task, but which probably, if people ask, so scientists, they will develop certain tools. And I hope that they will start understanding contributions, starting from microbiome in humans. So for instance, when it connected to health of people, this is what they care about.
Thank you. I have another question, but I wait for.
Nina, go ahead.
Hi, thanks for your talk. I just had, I hope, just a really quick yes or no clarifying question. So was the planarian secretory cell nidovirus discovered in 2018, or were you describing something that had already been discovered at that point?
Yeah, thank you for this question. So this virus was discovered. So this paper described discovery of this virus and comparative genomics. And so it is authored by the two teams, my group and also people who discover it is in Wisconsin. And so they discovered simply, it is also how the virus is discovered. So they sequenced all RNAs inside planarian cells, and then they assigned these RNAs to genome. And one RNA was unable to assign to genome. And this RNA is this virus.
Got it. Okay, interesting. And that was the largest known RNA virus at that time?
Yeah, it's still.
And it still is. Great.
It's still. I mean, it is what, and it is was actually was surprised to find this size. It's 41 kilobases because before, and so there is a figure in this paper which shows a dynamic of a change of larger genome size over the time, and it was like plateau. So people believed that viruses with RNA genomes larger than 35 kilobases doesn't exist. And so a discovery of this size genome, so previous figure one, if you could show figure one, Evan.
Figure one.
Yeah, so here you could see plateau. So this blue, it is over the years, it is coronaviruses basically. So all coronaviruses sequenced, they have did not exceed size of 33 kilobases, despite accumulation of genome sequences and despite the increase of genome size of DNA viruses, which was increased like several times. So people believe that it is limit to RNA genome size. So discovery was really surprising.
Great. Thank you so much.
I might add, I just really like this figure too, because you could see all these other coronaviruses. I had to look up, so I guess this is a virus that is in horses, and this is one that relatively recently emerged in mosquitoes that doesn't apparently have a pathological effect. But it seems like the knowledge is so fragmentary. Like how representative are these 4 genomes of coronavirus or nidovirus biodiversity?
So, sorry, in terms of size, it is covers the whole range of diversity from 12 kilobases to 41 kilobases. So it's these 4 viruses represent 4 families out of 15 families. So if you kind of put all other families, so they will be in between, and so they will have the same genome organization as these viruses. So excuse me. And, and that is
Time for more water.
not much talk. So virus is extremely diverse, and one of the challenges of analysis of the genome analysis, which I am talking about, was that you compare something that does not look like similar, and so you have to find very weak signals. And despite this very weak similarity, which we translate in certain functional structure, you could have seen at this figure that basic organization and so a resemblance between all the genomes. Size is different, but everything is similar. And that is shows that the virus is extremely conservative, and that is counterintuitive. So sequences looks like diverge extremely fast, and so, but there are constraints on certain features. So, for instance, order of genes is the same, and that is not true for humans. So, if you compare humans and mouse, similarity would be much higher between the two viruses, but genes will be kind of swapped in different orders. And so that is true for other organisms. So in viruses, in RNA viruses, there is huge constraint on change of the order. And on the other hand, there is a flexibility in terms of what could be replaced. And so replacement of nucleotides, it's going very fast and indeed, and many times. Yeah, so it's simply just kind of another message. So RNA virus is extremely conservative.
So thank you. So Raya had a follow-up question, and then Lyle has a phylogenetics question.
Mine is really quite a different question, but quite quickly, you can answer it or not. About being on war with the virus, that's something which is now a very dominant metaphor. I mean, I nearly collect them, right? So everything is about the war on virus. So would you say that in the middle of pandemic, we have to wage a war? Or would you say that even in the middle of the pandemic, we could adopt sort of more living with kind of approaches?
Yeah, okay, thank you for your question. I wish I know answer. Unfortunately, from one hand, a pandemic is the right moment when there is attention, right? And so then you could raise certain type of question and you could get attention and maybe could get funding, it could be some decisions and could go on. On the other hand, when pandemic and people suffer, it basically overrides everything else. It's very difficult to discuss about interest of viruses and kind of looking at viruses from kind of balanced perspective when goal is to kind of to stop pandemic. And so, so normally, I mean, if it's grassroots movement, of course you could kind of start this type of discussion. But from the government point of view, it is impossible, I think. So, I mean, people would not understand this, unfortunately. But discussing this type of questions outside pandemic, so there is no interest because viruses doesn't exist. If a pandemic, there is no pandemic, viruses doesn't exist.
Lyle, do you want to jump in with your question?
Sure, thanks, Ivan. So my question is about the phylogenetics. In your paper, I was seeing how you were kind of critically engaging with some of the phylogenetics claims made in the original SARS, at least early on in that period. So I guess I just have some basic question first about, could you explain some of the methods that are used to calculate the relatedness between viruses, and especially to calculate the clock of the divergence, evolutionary divergence that's used to make these claims about like SARS-CoV-2 emerged from its ancestor 40 years ago, 60 years ago. So what are some of the basic methods that are used and assumptions maybe that go into making those calculations? And then secondly, and this is just, I don't know if you have thought about this, but whether you have any kind of comments or critical perspective or on the ways that those kinds of calculations have been done for SARS-CoV-2 thus far.
Okay, yeah, yeah, thank you for your question. So if yeah, it is extremely important questions, and so they about how to visualize relationship between different viruses, and so to see order of bifurcation, and so and order of bifurcation. So how viruses diverge in which order. It helped us to understand origin of the viruses, and also put our knowledge in phylogenetic perspective, which is extremely important also for to rationalize our approach in the future. So when we talk about my work, which was done with SARS-CoV virus, major question was at that time not was immediate not immediate phylogeny, so how a virus emerged from bad viruses at the beginning. So question was kind of more larger, so whether this lineage was something absolutely new in coronaviruses, equal to our knowledge of all other viruses. So it formed a group on its own, very large group. So before it was known 3 groups, and like SARS-CoV formed a 4th group, and so that was proposed in the early papers. And so when we analyzed, we rejected this model, and we showed that this virus belongs to one of the groups already was recognized, and its representative, it was mouse hepatitis virus, which was the best studied coronavirus at the time. And coronavirologists welcomed this news very much because it provided also hope and also ability to project knowledge accumulated and study this virus using model. And why we arrived to different conclusions compared with other researchers, because I believe we used better techniques in terms of when you compare something. So to produce a tree, you need several steps. So one step, it is first you have to select what you compare with. And it's in this area, it's called alignment. So you have to align sequences. And so when you align sequences, so you use this alignment, and then you calculate distances or using other metrics to infer a tree. And so because we are in this field for 20 years before, we were able to produce better alignments at the time. And we used, and we were able to resolve better relationship between viruses. So if we kind of going to questions about origin of SARS-CoV or SARS-CoV-2, where it emerged like 20 years ago, 30 years ago, 100 years ago from bats, we're talking about small microevolution. What I told before, it was like macroevolution. And now we're talking microevolution. And so that it's different type of questions and also techniques, because in this case, alignment is not problem, but there are other problems there. So for instance, viruses could evolve not simply, so when they evolve, so evolution happens by replacements of nucleotides, so by mistakes. But also viruses use recombination. So they swap pieces of genome. They're called orthologous pieces. So piece which is responsible for the same function could be swapped between genomes. And if you don't account for this type of events, you will get completely different wrong trees. So you have to account for these events. And so there are special techniques how to do this. It's a complex techniques. So you have to extract from genomes certain areas where you could trust and build on this area tree. And since this has been done, and there is a paper from international group in Nature Microbiology where they were able to do this. And so they showed that they tested different type of models, and they showed that, okay, so you could have a confidence interval in terms of time of evolution, and so you could reconstruct basically character tree with certain confidence. I hope that it is kind of provides certain help.
Yeah, very good. Yeah, that's very interesting. So thank you very much. Yeah.
So thank you so much, Sasha, for your time. I'll follow up with an email with my protein modeling question. Great to see so many of you here. I really appreciate the intergenerational knowledge that's accumulated here, and all these rich archives of paper that you've shared with us. So I'm feeling like slowly but surely we're starting to understand what a coronavirus is. So thank you.
Thank you. Thank you.